Okay, so we're going to start the last session of the day. Arthur Dondi is going to start it with the talk named Single-Cell Transcript Isoform Sequencing for Precision Oncology.
So yes, and for precision oncology, especially in ovarian cancer and in the omentum. So what's the omentum? It's a sheet of fatty tissue covering abdominal organs that plays an immune function. It's basically a living barrier. Then it's the preferred site for ovarian cancer metastasis, and that's why it's of interest for us. So our goal in our study was to characterize the omentum on the full-length transcriptome single-cell level by sampling different tissue locations. So here 1, 2, and 3, for example, both healthy tissue locations and metastasis tissue locations. And the end goal will be also to discover neoepitopes and potential therapeutical targets in high-grade serous ovarian cancer cells. So to explain you a bit why we use long reads, let me first explain you what usually people do with short reads. So usually you have a tumor biopsy that then you isolate the cells and you capture all the RNA in order to amplify it and then to fragment it to sequence it on the Illumina machine to have short read sequencing. And from there, you can then compute what is the cell type of your different cells, and you can do many types of analysis, such as, for example, differential gene expression and pathway enrichment analysis, for example. Now, from the full-length cDNA, you can actually not fragment it, and you can have a part of the full-length cDNA that is used to actually add some PacBio primers that will enable you to sequence it, and to sequence instead of having short reads, so fragments of the full isoforms, you can then sequence the whole isoform. And this way, you can have the normal, most common transcripts. You can identify those. You can identify also alternative splicing, so different type of transcripts. You can also call mutations. And especially the most interesting for us was also to look at gene fusion. So, and transcript fusion here. And that's what we did. And this way, you have actually, for the same cell, you have the short read information and the long read information. With the long reads, you can basically have everything you have on the short read information, but on top of that, you have the full-length transcript information. The only drawback is that it's much more expensive, so that's why usually people don't do it. And it's quite recent. The first time it was done, it was in 2018. Now, to explain you a bit more the advantage of long reads versus short reads is that when you look at this data, so this is raw data from short reads, and you can see that here you have different possible isoform for one gene, and here you have the short reads. So the short reads are only covering some exons and sometimes even one exon of the whole isoform. So it's hard to directly determine which actual isoform is expressed. So here are all the possible isoforms expressed from this data. And you have methods that can infer which one are expressed, but you don't have direct observation of it. While when you use full-length cDNA sequencing, you can actually see which ones are expressed. And instead of having those, I think, 7 or 8 possibilities here, you have then only the 2 actual isoforms that are expressed. And here it will be so this one and this one under it. So that's why we actually wanted to have the full-length information. And then from there, you can actually, as you have more information than only the gene, because normally you will have, as you cannot really infer the isoforms, especially in single cell, from the short reads, you only have the reads like the gene information, gene level information. So when you had the full-length isoform information, the advantage is that you can, if you have the same depth coverage, then you can actually have a subclone and more information that emerge from this sequencing. Now, for our design, let's say, we had 3 patients because, as I said, it's very expensive. So we had only 3 patients, and we sequenced around 500 cells per patient. We have 5 samples. So for 2 patients, we had both the metastasis and healthy omentum samples, while for 1 patient, we only had the metastasis sample, no healthy one. And this is all patient merged, so like all samples together. That's the UMAP of the gene expression in short read. So this is Illumina sequencing. We basically did a PCA and then projected on UMAP. And basically, you can see here that, well, your cell types are determined through this way in short reads. And, well, you have the cell types that we have. We have mainly cancer cells, or AGSOC in violet, purple, sorry. Then we have a lot of immune cells, T cells, macrophages, and B cells. And then we have also endothelial cells and fibroblasts, and mesothelial cells also. And the interesting thing is that when you take then the long-read sequencing of the same cells, we projected the labels that were determined using the short reads. And we once again did a gene level on using the long reads this time, gene level UMAP projection. And you can see that you actually obtain the same clusters. So you can recreate with lower counts of cells because in long reads we have around 10K reads per cell, which compared to in RNA sequencing, usually you will have around 80,000 with short reads. So in our case, for long reads, we only have 10K, which can be considered as low, but it's also the highest count ever achieved for long reads single cell sequencing. And as you can see, you have the same clustering, so meaning that basically the data can be trusted as much as a short read. And when you go then to the transcript level, you still have the same clusters that emerge, meaning that while the information, is more sparse because instead of having the count for if you have 100 reads that actually are associated to one gene, then those 100 reads, if there are 10 isoforms in this gene, then they will be split between the isoforms. So, but even though the data is more sparse, we still have the same clusters that emerge. With some, when the cells are actually low count, they are low read count for the cells. You can see that you have some trajectories here, which are often associated to poor clustering. And I will probably in the future make some stronger filters on the read count for the cells in the first place to not have it, but I wanted also to show that you still have this kind of problem. And actually, Aiden told me yesterday that I should maybe also try not to use PCA for this type of data, as for the low count transcript, it becomes more or less binary, and then it doesn't work too much anymore. So now what type of isoforms do I have? Because when you look at the isoform reference, so the reference usually that is used is gen codes, well, when you look at the isoforms that you have in your sample, you can have multiple possibilities. You can have a full splice match that is an exact correspondence to the reference. You can have an incomplete splice match that basically corresponds to a same succession as in the reference of exons, but it just stops at some point because probably the read was streamed and the sequencing stopped. You can also have a novel in catalog, meaning that the in catalog means that you have something that is not in the reference, but it uses junctions, exon-intron junctions, that are actually known from the reference. And you also can have a novel not in catalog that uses actually junctions that are not repertoried. And for example, here it can be an intron retention, something like that. Now, once I explain what are those type categories of isoforms, now in my different cell types, I looked very generally at what type of isoforms I had. And I was surprised that, first of all, in all the cells, you have this repartition of the isoforms if you only look at novel in catalog, novel not in catalog, and full splice match. Because usually the incomplete splice ones, you want, as you don't know which one they are, because it could be something like a new one, something that doesn't correspond to the reference, or it could be just a full splice match that is incomplete. So after discarding those and looking only at those 3 types, you can see that actually you have a high part of your data that is full splice match, but you also have more than 30% of the isoforms that are actually novel ones. And here we had around 500,000 isoforms total, meaning that you have thousands, hundreds of thousands of new isoforms that are unraveled this way. And when you look at the distribution in different cell types, you can see that cancer cells are the ones with the less full splice match, so they are the ones with the most new isoforms. And well, it's quite expected, as you expect cancer cells to have more isoform variability, more expression type of isoforms in general, because first of all, cancer cells, they express a lot more isoforms and reads in general. So that's quite expected. But when you look also at the support, because those new isoforms, people can ask me, what tells you that those new isoforms are true or not? Because it's hard to say. Well, first of all, for the reference, when you look at how it was built, it's not that they just found things and they report it, and that's how it was done. Like, there is not much more categorization with the free end. Is there any free prime end support or 5' end support? But still, I wanted to do it because I wanted to see how much of those new isoforms they actually end up nearby. For example, for the 3' end support, how much of them ends nearby a poly A site, and how much of my isoforms end up nearby a cage peak site. So the cage peak site is basically a start of transcription site. And you can see that in both novel not in catalog, novel in catalog, and full splice match, we have around 80%, 70 to 80% of support for those new isoforms and even known isoforms, because not all known isoforms actually end up nearby those poly A site and cage peak sites. Those both are actually sites that were determined experimentally, while some of the known isoforms were never validated. So that's also why you actually end up with not all the known isoforms being nearby poly A site or cage site. Now, if you look at, we determined some type of classification for the isoforms because we were interested to know which of the isoforms are only present in one cell with sulfur efficient coverage. So we took every isoform that were so at least 5 times. And we looked at which one are actually present in only one cell, which one are only present in one cell type, and which one are present in only one sample, so one patient. And the results are here. And in percentage of the isoforms found, you can see that in cancer, once again, you have much more of the cell type specific isoforms. So meaning that cancer samples, they actually share much more isoforms than the other cell types. And same for the isoforms that are only present in one cell. You have much more isoforms that are only present in one cell, because, as I said, those cells express more things and they have more variability in expression. So that's basically it for this. And in terms of sample specific, and basically just to explain a bit maybe more for the cell specific, you can see that very few of them are cell specific. So this is basically a zoom in percentage here, and that's the raw count here, because you don't have the same counts for the different cell types. So I wanted also to show that it's supported both in terms of percentage and in terms of counts. Now, another thing that was of interest for us was the fusions. So which of our isoforms actually spawns on 2 very far away and different genes that can be on the same chromosome or on different chromosomes. And this is one example of one that we found that I wanted to share with you to show what the result can look like.
After 1 minute.
Yes?
1 minute.
Yes, okay. And basically, then maybe I will skip what this fusion is and just show you on how you can look at it. So basically, you have here the expression profiles of for one gene. Here is IGF2BP2. And these are all the reads that you can find. And here you are only the reads that are fusions. And you can see that the gene starts here, and when you go through there, it actually stops, because on this stage, the read will then cover another gene, and it will just basically be a fusion where there is actually a genomic breakage happening between those two exons. And well, you have just then, chromosome 3 and chromosome 11 fusion. And just I wanted to show you this genomic browser picture, also to just show, as a last thing maybe, that the short reads, you can see that this is the expression patterns of the short reads compared to the long reads. And the long reads, you can see that they correspond perfectly to the exons of the genes, while for the short reads, as you have a lot of trash data that you cannot filter because you don't know what it is, you basically have this type of very noisy data in single cell that makes it quite hard actually to interpret anything in terms of isoform expression. You only can focus on basically the whole gene expression. So basically you take all the reads that correspond to an exon and you add up to make the gene expression. And that's, well, that was just an example to show you a bit how the long read will actually completely correspond to the isoforms expected compared to the short reads. And that's it for me. Thanks a lot.
Thank you, Arthur. Do we have any questions from the audience?
Yes, Jens?
Jens, go ahead.
Okay. So I mean, I would guess that many of these are not functional, right? So they're spliced in a way that makes them non-functional.
So you mean for the fusions?
No.
Even before?
I mean, the fusions,
The isoforms.
many of them are probably non-functional, right? But some are really functional.
For the fusion, it's true that, for example, this one.
If you go to I mean, sort of what could be, what is a transcript from one gene, but it's spliced in a different way than what you would expect, and especially if it's only found in one cell, most likely that is not the functional variant, right?
I wouldn't assume that. Well, first of all, from the transcript itself, you can actually look at it like you can predict if it's functional or not. That's actually something I'm doing right now those days. And while I don't have the final results, no, that's not an assumption that stands for the moment, at least.
So there was a discussion. I mean, the protein folding guys were quite interested in this for a while, and saying that many of the splicing variants are not functional, etc. So if a fair portion of these are not functional, it simply means that you have downregulated that gene, right? And if you don't have this information, it may look like it's upregulated or on a normal level, etc. So I think it's very interesting if you go ahead and do that analysis of whether they are potentially functional or not,
Yes.
and then sort of correct.
Yes, absolutely. That's actually, like, I would say, the last piece of the puzzle when you look at this on this aspect. And, well, at least for what I found, there are less functional, or at least less functions that are found in the novel ones, but you still have a fair portion. Yes.
Two things. I'm just trying to think, is there a way to computationally predict, you know, some of these fusions, or to do something that might, like, I don't know, help here? And there's two things, like, when the folks do mass spec analysis, like the whole genome mass spec, they identify tons of proteins that aren't like in gen code. Yeah. And a lot of the times those are just thrown out because they're not in gen code and they don't know what to do with them. Yeah,
and most of the time also the mass spec is actually template based, right? So you don't even look at them.
No, the whole genome mass, or the whole proteome mass spec isn't.
No, but
just doing all the fragments that they can do. But a lot of the stuff is thrown out because they can't map it to the reference genome. So like these are proteins that are observed, but they're not like they're not in gen code.
Yes.
So that's maybe one thing that could actually be done is actually, you know, doing a T-blast in of basically all the protein signatures from the mass spec and basically doing it against this, and that might give you some information if those are expressed. The second thing that might be interesting is if you take the TCR data, and there's like tons of T-cell receptor data now that's being generated by a lot of labs in cancer. People thought that the antigens that would be observed would be these neoantigens associated with mutations.
Mm-hmm.
And when you do the searches against the databases, again, most of those neoantigens are not being detected. So we have like all of these peptide sequences from the TCR sequencing, which these T cells are recognizing. But by and large, if you map 10% of them to known antigens, whether they be viral, bacterial, or predicted mutations, the rest of them you can't map.
Yeah.
So this would be, those would be two really quick, relatively quick things to do. You know, take all of the sequences.
I actually have the mass spec, some mass spec information for those samples. Not the same cells, of course, but I have it, and I started also looking at it. But well, that's not the easiest thing to actually correlate it. There are other teams that are working on it. And but yeah, that's also part of the next step. Yeah, definitely.
So let's give another round of applause.
Thanks.